So to continue with Edmond degradation, I just want to let you know how this intermediate, unstable intermediate forms is from the thiol group attacking the C double bond O of the amide, the first amide. So it cyclizes at this carbon, making this cyclic intermediate. And your book on page 1203 has a great mechanism written out if you're curious. I'm not having you be responsible for it, for the mechanism, but I just want you to know that the phenyl isothiocyanate basically labels the N-terminal amino acid. This condition in which it cleaves this, it will only happen to that amino acid that got this phenyl isothiocyanate attached to it, because it forms this 5-member ring, and then while it's forming the 5-member ring, that's how it cleaves this off, and this becomes a leaving group over here, and that leaving group is basically one less amino acid, and the rest of the peptide is attached to this. Okay, so because the R group is going to be different for every amino acid, this is how the identity of the amino acid is determined, all right, by looking at what the R group is. So you could repeat this. So you repeat the process. And this Edmond degradation is now been fully automated. So there's an instrument that does this automatically, and it could be done any small peptide less than 50 amino acids. This method is the method of choice for doing this. So if you started with 50 amino acid protein, you do it one pass at it, there's 49 amino acid left. You do it again, and then there's 48, and you keep repeating it, and then you keep getting the identity of the amino acid, and once you're done, then you're done. And this is how Meganin 1 and 2, the sequence has been discovered, because this is only 23 amino acids, and Edmond degradation would be the ideal way to do this. Okay, now for peptides that has more than 50 amino acids, so much bigger proteins, this is not a practical way to do it. So the method of choice for anything bigger than 50 amino acids is to use enzymes. All right, so we take advantage of the fact that there are peptidases. So those enzymes are enzymes that cleave the amide bonds, and they're very, very specific. All right, so I'm just gonna go over two peptidase that your book introduces. One is called trypsin, and trypsin is very, very specific in that it only cleaves at the carboxyl side of lysine and arginine. So these are the basic amino acids, as you recall, right? So what I mean by carboxyl side is if you have this Meganin 1, and if you treat this protein with trypsin, then on the right side is the carboxyl side. On the left side would be the amino side, because this is N-terminal and this is the C-terminal, right? So after the lysine over here on the right-hand side, it will cleave. And then, let's see, there's another lysine, so it will cleave there. It will cleave here. Are there any arginines? No. So, Meganin 1 would have 1, 2, 3, 4, 5 fragments from this treatment. All right, so remember that is on the carboxyl side, right? And the second enzyme is chymotrypsin. And this cleaves at also the carboxyl side of phenylalanine, tyrosine, and tryptophan. And these are the amino acids with aromatic benzene ring. In the side chain. All right. So you have to kind of keep these in mind, where it cleaves and how specific they are, and then using this, you could determine the sequence of peptide. Okay. Now I'm gonna go over the skill builder problem in 1204 with you. But after we go over that, I expect you to be able to do 25.31 on your own. This it's not a big protein that you are sequencing. They give you something small to practice. But this is basically, you have a long chain. So think of protein as a long chain. And depending on which enzyme you use, is going to cleave at different sites. So let's say this is cleaved with the chymotrypsin, and these are you get 4 fragments, and then you take the same protein, but you treat it with enzyme B, and it will cleave at different places. All right. Now you have the results from here with 4 fragments, and you have results here 3 fragments. But notice that they are cleaving at different places. So then what you try to do, figure out, is using both information, if you could try to figure out, see where it's cleaved here. Here is not cleaved, so that gives you the sequence here, right? Same thing as here. This is cleaved here, but here they're intact. So you could try to figure out between the two things the whole sequence of the protein. All right, so let me just briefly go over this problem that's in your book. And this is in page 1204, Skill Builder. And what it's asking you to do is, you have a peptide with 16 amino acids, and it's treated with trypsin. So that's one experiment. And then they took the same protein and then treated with chymotrypsin. All right, so 2 different experiments. And you're asked to find the sequence of the 16 amino acid. All right. So what you're doing here is, you know that trypsin cleaves at the carboxyl side of lysine and arginine. So out of these fragments, there's going to be, so it's how this fragment has lysine and this fragment has arginine. So that means that it was in the chain. And notice that this proline with cysteine is the fragment that doesn't have arginine or lysine. So that means that this fragment probably was the one that had the C-terminus of the original peptide, right? So we zoom in on that, and we know then that the cysteine is the C-terminus amino acid of the protein. So that's all the way out on the right hand side, and then we know that proline is right next to that. So the question is, next to proline on this side, does this fragment go in here, or does this fragment go in here? There are 2 choices, right? It could be either alanine and this fragment over here, which means that this fragment is the other fragment in the beginning. So, and then the other choice is this fragment was the one next to the proline, which means that this fragment was the one that was beginning. Do you follow me? Right? So in order to answer that question, then you look for a fragment that has proline and cysteine to see what is the next to the proline. So notice on the chymotrypsin fragment, you have the proline and cysteine right here. And notice this lysine that is next to it, right? So that means that this fragment right here is the one that is immediately preceding the proline. So take that fragment and put it here, which means that this fragment right here has to be the one that is beginning, which means that isoleucine is the N-terminal amino acid. All right, so that's how you would solve this puzzle. And notice that's the solution here. And I would hope that, you know, using the same strategy, that you could apply that to 25.31. And if you have any questions and you can't do it, or at least try it, please. And then if you can't do it, write down the specific question as to why you couldn't solve it. All right, what were you stuck on? And then we'll go over it in class. All right, so that concludes this lecture.